Ecological diversity and agriculture. The objective of these things is ecological diversity, wildlife and agriculture, GM crops and their impacts on the environment, insects and agriculture, pollination, the forest fragmentation and agricultural biodiversity concerns. So GM crops is a genetic modified crops which have been generated from a DNA trimmed option or DNA segmented things to improve and make the needs to be makes the demand to be needed. Inserting a gene into a cell collectively, these techniques known as a recombination of DNA technology. In India, it is most one of the thing. Most of the crops which have been burned because of this genetic modified organism GMO. So this is one of the important topics of our subject. Genetic modified technology allows the transfer of the genes specific traits between the species using laboratory techniques. GM crops were first introduced in the US mid 1990s. Most current GM crops used in the are engineered for the insect resistance or herbicide. GM crops grows in a correlate includes corn, alfalfa, sugar, beet, soya bean and the colon. Potentially future applications of the technology includes nutritional enhancement, stress tolerance. GM crops are regulated at the federal level by the US Department of Agriculture. Examples in food crop resistance to the certain pest diseases, environmental conditions, reduction of spoilage, resistance of chemical treatments, improving the nutrition profile of the growth. Genetically, crops has been gene added or removed. Genetically engineering techniques are originally introducing gene and guns, electroporation, microinjection, and agrobacteria. Gene guns, also known as the biologist, shoots direct energy to the practical radiations and against target genes into the plant cell. The most common methods DNA is bound to be tiny particles of gold or a tungsten, which is substance shot into a plant tissues or single plant cells under the high pressure. Agrobacterism, medicated transformation, also common as technique. Electroporation is used for the plant tissues, does not contain cell walls in this technique. DNA enters the plant cells through the miniature pores which are temporarily caused by the electric pulses. Transgenic plants have a genes inserted into the derived. This is the plant which have already been transgenic. Between the kingdoms, it inserted DNA has been modified slightly in order to correctly affect the host organism. Cisgeny plants are made using the gene found whether some species or closely related one where conventional plant breeding can occur. Subgenic genetically modified plants can also be developed using gene knockback or the gene knockout. The impacts of the powerful scientific techniques caused a dramatic expansion of genetically modified crops leading to altered agricultural practice posing direct and indirect environmental implications despite the enhanced yield potential risk and the biosafety concerns. Thank you.